AS Level Biology 9700. Paper 1, Variant 3, October, November 2024. Students, you know, already we have discussed question number 1 to 20 into 2 videos. Now, this is the third video to discuss question number 21 to 30. So, we will start from question number 21, and you can see on screen a nucleic acid molecule has been shown over here, and this nucleic acid molecules contain uracil. It means this is RNA. So four nitrogen bases have been shown over here, and on the other hand, a phosphate molecule and sugar has been shown in the form of backbone of this nucleic acid molecule. So they have asked about what is the name of bond X. So you can see the bond X is formed between phosphate group and sugar molecule. Plus sugar molecule. So you can see over here from the first option, glycosidic bond. And students, you know, the glycosidic bond is formed between a nitrogen base or sugar molecule. Or secondly, sugar molecule. This is the plus nitrogen base. Secondly, the glycosidic bond is formed between two sugar molecules to form the disaccharides or polysaccharide molecules. So glycosidic bond cannot fit over here. The next is hydrogen bond. Hydrogen bonds are formed between the nitrogen bases, which are very weak bonds. So this bond is not there. The peptide bond is formed between the protein molecules or in amino acids. They join together with the help of peptide bonds. So phosphodiester bond is left behind. So the phosphodiester bonds are formed between the phosphate molecule, and 5 carbon of one sugar molecule and 3 carbon of another sugar molecule with the phosphate group. So according to this, you can see this molecule is the phosphodiester bond. So D is the correct option. Moving towards the next question. Question number 22 has been shown over here. XNA is a laboratory made nucleic acid. XNA is made of nucleotides in which one component has been replaced by chemical X. The chemical X is organic but is not found in nature. Organic but not found in nature. Organic. Focus on organic. The part of the molecule responsible for coding is not changed. It means the part of the molecule responsible for coding is not changed. And coding part of the molecule are nitrogen bases. So the nitrogen bases, purines and pyrimidines, they cannot be the part of this nucleic acid. So in this way, C and D options will not be the correct options. So now 2 are left behind. 5 carbon sugar, which is an organic molecule, and phosphate group is inorganic molecule. So it means A is the correct option. A is the correct option. They have told us that which component of DNA or RNA nucleotide has been replaced by the organic chemical X. So if there is change, the sugar molecule is part of the backbone sugar, but it does not directly carry the genetic code. So modifying the sugar would change the backbone, but not necessarily the coding potential of the bases are still able to pair correctly. So it means the organic molecule that can be changed, and it will not bring about any change into the coding pattern or coding sequence. So it is the carbon molecule. But if the phosphate group is changed, so modification to the phosphate group would alter the backbone. So secondly, it is an inorganic molecule again. So only A is the correct option, which is a 5-carbon sugar. Next question, which strand of DNA does RNA polymerase bind to? So lagging strand and leading strand. You can see the lagging strand and leading strands are the cannot be the correct options. Why they are not correct options? Because the DNA does RNA polymerase bind to. So DNA and RNA. So in this way, we can see that the lagging strand specific to the DNA replication. So lagging and leading strands, it takes place during the DNA replication. So the lagging and leading strands are terms which are used in DNA replication, not in transcription. So in DNA replication, DNA polymerase synthesizes DNA on both strands, but in different manners due to anti-parallel nature of DNA and the 5 to 3 end synthesis requirement. So this option is irrelevant to transcription. So these first two options cannot be the correct options. Now the third option is which is called as non-transcribed strand. Non-transcribed strand, also known as the coding strand. So RNA polymerase does not bind to the bind to and transcribe this strand directly. So RNA molecules synthesized will have a sequence almost identical to the non-transcribed strand. So it will not be the correct option. Now the last option is left over behind, that is the template strand. So this is the correct term. Why this is correct term? Because the RNA polymerase binds to the promoter region of the DNA and then moves along the template strand, and then it using its sequence as a guide to synthesize the new RNA molecule. So I would like to say that RNA polymerase binds to the template strand of DNA. So D is the correct option in this case. Now question number 24. Which processes occur during the formation of messenger RNA? So the formation of messenger RNA. They have given us the first of all the condensation formation of messenger RNA. So condensation is a reaction where smaller molecules join together to form a larger molecule, and when they join together there will be a removal of water molecule. In the synthesis of RNA, nucleotides are linked together by phosphodiester bonds. So the formation of each phosphodiester bond involves the removal of water molecules. So the condensation can be involved in this case in the formation of messenger RNA. Second is polymerization. Polymerization is the process of joining many small molecules or monomers to form a long-chain polymer. In the case of messenger RNA synthesis, individual ribonucleotides are joined together to form a long messenger RNA strand. So this is called as polymerization. So it can also be the part of the formation of messenger RNA. So the third is replication. Replication is, the process of synthesizing a new DNA molecule from an existing DNA molecule, or copying of the DNA molecule from the existing DNA strand. So replication is not the formation, it is actually the copying of the formation of messenger RNA. So this is not the part of this, you can say, formation. So the last is transcription. Transcription is the process by which the genetic information in a strand of DNA is copied into a new molecule of messenger RNA. So this is the primary process for messenger RNA formation. So condensation also occur, and then you can see condensation occur, polymerization occur, and transcription occur. So according to this, you can see the B is the correct option. 1, 2, and 4 are the correct options. So correct statement. So B is the correct option, in which 1, 2, and 4 processes are involved in the formation of messenger RNA. Question number 25. The anticodon for the amino acid tryptophan is ACC. Adenine, cytosine, cytosine. So that shows a template DNA sequence that includes the DNA triplet for tryptophan. So we have to show a triplet. So uracil, guanine, guanine. So this is the first option which they have given in the first option. You can see uracil, guanine, guanine, a pair. Adenine always pairs with uracil. Adenine always pair with thymine, but in RNA, adenine always pair with uracil, guanine with cytosine. So this is the combination in which they make pairing with one another. So complementary messenger RNA UGG pair with uracil, cytosine pairs with G, and C pairs with G. So messenger RNA codon to template DNA triplet. So the messenger RNA codon is transcribed from the triplet DNA strand. So therefore the messenger RNA code is complementary to the template DNA triplet. So given messenger RNA codon, uracil, guanine, 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 uh, this whole cannot contains A, C, C. So it cannot be the part of the anticodon for the amino acid tryptophan. Again, in the second, there is no adenine, cytosine, cytosine. Again, there is no, but here adenine, cytosine, cytosine is there. It means D is the correct option. B is the correct option, in which adenine, cytosine, or ACC is present in this triplet DNA sequence. Next, question number 26. A student draw a plant diagram of some plant tissues. You can see over here, the diagram contains a mistake in the way that it is drawn. So the maximum width of R is 15 millimeter on the diagram, and its actual width is 250 micrometer. The student was asked to draw a scale bar on their diagram. So here you can see part of the plant drawn. You can see this arrangement shows, in a root, there is star-shaped xylem, and phloem is there. But this is stem. The figure or diagram shows that this is stem. So first two options are not correct. Now, Both of they can be the stems. We have to find out one. So we have to calculate length of the scale bar in millimeter to correctly represent 100 micrometer. For this purpose, we will first of all 15 millimeter on the diagram, and the actual length is 250 micrometer. So magnification is equal to width, maximum width is 15 millimeter, and actual length is 250 micrometer. So first of all, we will determine the magnification will be equal to width over actual. So width is 15 millimeter. We have to convert this millimeter into micrometer. So 50, we will multiply 15 with 1000. So it will become 15,000 divided by 250. So it will be divided by 250. When we divide, it will become equal to 60. So the diagram has a magnification of 60x. This means every 1 micrometer is actual size is represented by 60 micrometer or 0.06 millimeter. So calculate the length of the scale bar for 100 micrometer. So it will be equal to, in this way, length of scale bar, length of scale bar is equal to 100 micrometer into 60. So it will be equal to 6000 micrometer. We will change it into millimeter. Then it will be 6000 divided by 1000. It will be 6 millimeter. So according to this, you can see the D is the correct option, but there is a mistake. Xylem, or there should be phloem, because this is phloem, not xylem. So D is the correct option. Question number 27. 4 students have drawn and labeled part of a structure seen on the electron micrograph A, B, C, D. So first of all, we will go with the A. They have shown us the sieve plate, a thin cytoplasm, and mitochondria. Mitochondria gives the energy, sieve plate, and the thin cytoplasm. It means they are part of the sieve tube elements. Vacuole is not present in the sieve tube element, and this is not the place where the vacuole should be. So chloroplast is also not there. So B and D are minus from the correct options. On the other hand, you can see the option C and D remained behind. So in diagram C, you can see the label nucleus within the sieve tube. This is definitely incorrect because mature sieve tube elements are without nucleus. So this is also not correct. So only A is the correct option, in which the mitochondria is there. Few mitochondria are there to give it energy, and thin cytoplasm is there, which becomes on one side. So this is A is the correct option over here. Now moving towards the next question, which is 28, and question number 28 is the diagram shows some structures used for transport in the phloem. Cell Y, companion cells, direction of transport, or cell Z. Which statement is correct? Cell Y is the sink cell. Sink, sink is that cell where the food has to reach from the source. Source are those points where the food is manufactured. So Y is not a sink, it can be a source because it is present beside the companion cell. Also, food will be manufactured in the cell Y, so it is a source cells, it is not a sink cell. Food, or it can be a mesophyll cell of the leaf where the food is manufactured. Then food has to move towards through the companion cells into the sieve tube elements where it has to move towards the other parts of the plant where towards the sink. So second is cell Z can be above or below cell Y in the plant. So cell Z can be a sink cell. So due to which it can be above, or because the food transportation takes place in both directions. It can move in the upward direction, so it can move in the downward direction. So uh, it can be in the upward or downward direction. Cell must be in a photosynthetic tissue. No, it is not present in photosynthetic tissue. It is present in the downward side. So it is not the photosynthetic tissue. So cell Y allow glucose to move to the sieve tube via the companion cell. The last option D is the last option is D, which is according to which we have to choose whether it is correct or not correct. So cell Y represent, as I have told you, that it represents as a, so these sugars produced in source cells are primarily sucrose, not glucose, for transport in the phloem. So glucose is typically converted to sucrose before phloem loading. So food is not transported in the form of glucose, it is formed in the sucrose. So this is not the correct option, only B is the correct option. Question number 29. So question number 29 is, the diagram shows a section through root of a dicotyledonous plant. Which statement correctly describes the movement of water and solutes through this root? So soil particles, root hair cell, root cell, and Casparian strip and xylem vessel. A layer of which statement correctly describes the movement of water and solutes through this root? So a layer of Severian cells causes water and solutes to move from the apoplast pathway into the symplast pathway. So the difference is between these two types of pathways, the symplast and apoplast pathway. Apoplast pathway, when the water and solutes move through the cell walls and intracellular spaces without entering the cytoplasm of the cell. It means that it will not disturb the cytoplasm because it is very faster speed, because less time will be to move the from. So layer of Severian causes water and solutes to move from the apoplast pathway and symplast pathway when the water has to, on other solvents has solutes has to move from cell to cell through cytoplasm, and when there is suberin causes. So Casparian strip is a band of suberin, a waterproof waxy substance is there which is embedded in the cell walls of the endodermal cells. So its primary function is to block the apoplast pathway. So if the pathway is blast, then the apoplast pathway is changed into the symplast pathway. So this can be the correct. So layer of Siberian causes water and solutes to move symplast pathway into the apoplast. It is opposite of the first statement, so it is not correct. The third statement, the water has moved from the symplast pathway into the apotonoplast, will will not causes water and solutes to move from the symplast or apoplast. So last C and D are not the correct options. Only one correct statement is, which is A, because only or when there is variant due to the Casparian streak, the what will happen? There will be the blockage of the water, so water will not move through cell wall. So instead, it has to take the path, symplastic pathway. It has to pass through the cytoplasm. So according to this, A is the correct option. Next question number 30, and question number 30, you can see is a graph. The graph shows the effect of different partial pressures of carbon dioxide on the oxygen dissociation curve for hemoglobin. So here you can see partial pressure of carbon dioxide, which is 1 kilopascals, and partial pressure of carbon dioxide is 1.5 kilopascals. So percentage saturation of hemoglobin with oxygen has been shown. These are two curves. What is the change in percentage oxygen saturation of hemoglobin at a partial pressure of oxygen of 6 kilopascals as the partial pressure of carbon dioxide changes from 1 kilopascal to 1.5 kilopascal? So find out the correct option. We have to go with this graph. First of all, we will move towards the 6 kilopascal. So 6 kilopascal is there. This is the 6 kilopascal. So first of all, with at 1 kilopascal, it is about 65, and then on this curve, so you can see over here, reading is 65% saturation, and then it becomes less. It is about 6, little bit less than this. Let me draw it again. It is less than this. You know, it is very difficult to draw on screen, but you can, so it is about 45. So change is equal to final saturation minus initial so change is 45 minus 65, so it will be equal to minus 20. So, you know, there is difference in the size of these pages which we use on screen and the other pages which are in examination and due to according to the marking scheme. So this is the method. How can we find out the change uh, of this. So this is, I have given you the simple method. So according to this method, you can see this is the answer, which is B. So this is minus 20%. So 20%, so it is the answer. So you can find out in examination, you can find out in this way. Maybe the results are little bit changed, the lines drawn are little bit changed, but this actual method is this. So it is all about from question number 21 to 30. Please also watch my previous two videos on this paper and wait for the fourth video in which I will discuss from question number 31 to question number 40. And please don't forget to like, share and subscribe my channel. Thanks for watching my videos.